signal shows up, then I take the enzyme, split it, and the active component, now this is the active signal, can bind to the protein. And now I can activate this protein. And this is the behavior that's going to be expressed by the cell. And the question, of course, is, well, what really activated this protein? What was the original source? The answer is the primary signal at the environment was then relayed by the secondary signal to activate the behavior of the cell. So without all the labels, if we just quickly look at it, let's just go right through it real fast. And the point about it is this. Let's see. That here's the environmental signal. Here's the receptor, the effector. That's the perception unit. Perception is now being started because it saw the environment, changed the shape, activates the enzyme. The enzyme is activated, breaks that molecule, the signal molecule. The signal molecule comes down, binds to the protein, and generates behavior. The bottom line was this. The behavior of the cell is not programmed. The behavior of the cell is continuously adjusting to whatever the signals are in the environment. So now I got another question to ask you. What happens if that environmental signal shows up, but I don't have the proteins necessary in the cell right now for that event? So when it shows up, it says, oh, I can't make a response. I don't have the behavioral proteins. Where do we get the behavioral proteins? Now we bring the DNA back in. What's the role of the DNA? The DNA double helix actually is a blueprint of the protein. If I separate the helix into two separate strands, and you look at these are called bases, these are the steps of the double helix right here, the color sequence in the DNA codes for the sequence of the amino acids. So for every 3 bases, I can tell you which is the next amino acid. So the point is this. The plan of how to make a protein, a specific protein, is built into this DNA. So that every 3 bases say, oh, put in tryptamine. Okay, put in proline. Next one, put in alanine. Whatever it is. So the sequence, the DNA is a blueprint for the protein. Okay? That's all it is. It doesn't have any action except when I need it. So how do I activate the gene? Well, you heard of cancer genes? And you say, what is that, a gene that gives you cancer? And the answer is, here's a simple truth. Genes do not self-activate. That's biochemically a truth, meaning a gene cannot turn itself on and a gene cannot turn itself off. If you want a gene to be active, it's not up to the gene. So the concept that there's a cancer gene is a false concept, meaning this. If the gene really caused cancer and you were in possession of that gene, when would you express the cancer? You would express it by the time you were born. Because as soon as the cells started to divide, the cancer gene would say, okay, time to make cancer. So how can you have so-called cancer gene for 30 or 40 years sitting in your body and you don't have cancer and then you get cancer? Should I go back and say the gene caused that? And the answer is no. In this paper by Niehaut, Metaphors in the Role of Genes in Development, he played it out in this a simple statement of truth that I'm going to show you because I want to use his paper because the statement is so perfect. But the fact is this. What did he say? He said this. For 50 years, we have believed that genes are in control. We've been repeating it and saying it over and over again for 50 years so that it's part of every textbook. And the bottom line was that was never a scientific reality. It was never scientifically established that genes control anything. It's not true. What is the truth? Well, the answer is this. The first thing, conventional belief, genes control biology, is totally false. Why? Genes can't turn themselves on. Genes can't turn themselves off. How are they going to control anything? They can't control themselves. So bottom line is the genes aren't in charge. So the question is, if I need a gene to be activated, and why would a gene be activated? To make the proteins for the cell that needs to do the behavior. So the truth statement is this. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of the gene. Well, that's somewhat of a complicated sentence, so let's simplify it. Just read line 2 and line 4. And if we read that, it says, a signal from its environment activates expression of the gene. What does that mean? The genes in your body are selected not because they're self-selecting. The genes are always selected in response to the environment that you're in. So if you had that cancer gene and for 35 years, let's say, you stood around saying, hey, I don't have cancer, and all of a sudden cancer happened, are we going to go to the gene and blame the gene, or what would we actually look for as responsible if you understand the true statement? The signal from the environment. What change in your life promoted activation of the gene that was sitting dormant for 35 years? We've been focusing on the gene all the time. The point is, we have to start focusing on the signals. The signals do this. Well, let me explain how this happens. When we look at a cell nucleus, this is where the chromosomes and the DNA is. I can stain the chromosomes, and you break open the nucleus, you can see all these different chromosomes. You have 23 pairs of chromosomes to make a human. They're called pairs of chromosomes because you get 23 chromosomes from your mother, 23 chromosomes from your father, and they're matched pairs. So, in fact, even I can see that one of these is from the mother and one of these is from the father because you can color code them. Well, that's a nice, interesting experiment, but the question is this: what am I staining? I'm not staining DNA. So what's in the nucleus? And the answer is this: 50% of the nucleus is DNA and 50% is protein. And the reason why we have a problem here is this: for 50 years, everyone was so focusing on the genes that when they wanted to study the DNA, what did they do? They'd go find a nucleus from the cell, they'd break it open, expose all the chromosomes, and you know what they do? Separate the protein from the DNA and then throw away the protein. And for 50 years, they've thrown away the protein in their focus on studying DNA. And now, all of a sudden, in the last few years, the question is, hey, what have we been throwing away? And the answer is the control. For 50 years, they've thrown the control away, what controls the genes, and studied pure DNA. There is no such thing as pure DNA in any organism. The DNA is always associated with the protein. So what's the function of the protein? Look how simple this is. The protein forms a sleeve around the DNA. So let me give you an example of what I mean by that. Imagine my bare arm is a gene. Let's say it's a gene for blue eyes. And I say, okay, can you read the gene for blue eyes, yes or no? Yes. Okay, but all of a sudden, well, what does the DNA look like when I put it back in the nucleus? Put the sleeve of protein on it. Can you read the gene for blue eyes, yes or no? If you want to read the gene for blue eyes, what do you have to do? Take the sleeve off. Well, the sleeve is protein. How does a sleeve come off? Here's the protein, and it's locked onto my arm. If you can remember back about 15 minutes ago, what is it that will cause the change in the shape of the protein? So when I add the signal from the environment, all of a sudden, then what happens is this: the protein changes shape, pulls away from the DNA. Now I can read the gene, and when the signal is removed, the protein will come back and cover up the sleeve again. So the bottom line was this: the gene was just sitting there all the time. It's whether the proteins are present or absent. So if I look at it this way, then we understand this. I said you were made out of protein. The understanding is that DNA is the blueprint for the protein. In conventional textbooks, because they've thrown away the protein for 50 years, they don't talk about this. Conventional talks about the DNA goes to the RNA, which is like a Xerox copy of the DNA, and the RNA is then turned into the protein. And then they talk about the primacy of DNA. That's what's in all the textbooks. You are a result of your DNA. But they've thrown away the protein. So when we put it back in, it says, the protein covers up the DNA. The protein is a sleeve. But to take the sleeve off, you actually have to have the environmental signal. So remember what Niehaut's quote was? A signal from the environment activates the expression of the DNA. So the bottom line right here is this: the environmental signal comes in and changes the shape of the regulatory protein, which removes the sleeve, exposes the DNA, and then I can make my proteins. So rather than the primacy of DNA, which is conventional thought, it's actually the primacy of the environment. It's the environment that selects your genes, not the genes themselves. So if I wanted to illustrate it, let's go back to our picture of how the cell worked. What I showed you